One of the most abundant industrial side streams in the world is of course CO2. Using this carbon source efficiently instead of pumping it into our atmosphere is no doubt one of the most important areas where synthetic biology can help. The quest for microbes naturally capable of using this abundant raw material takes us to the very beginning of our planet. At Aalto University, Professor Silvan Scheller's team is working with an intriguing group of microbes, methane-producing archaea.
Question. So they actually come from the time before there was oxygen on our planet, and they probably originate even before photosynthesis. And so this was actually a time where there was much more CO2 in the atmosphere than what we have now. And these organisms, they have learned to actually eat the CO2 and then with the help of hydrogen convert that CO2 to methane. And basically by producing a fuel, they can extract energy from that process. And so that's quite remarkable. And so nowadays you find those at sewage plants or in rice fields or in the rumens of cows, or a few inside you maybe also. So they are at places where there's no oxygen because they're actually oxygen sensitive. And that's actually why we use this laboratory equipment where we work with the exclusion of oxygen, so to keep them in their environment that they are able to live, basically.
These organisms could serve as a microbial alternative to the Fischer-Tropsch synthesis in a Power-to-X plant.
In the future, the microorganism can also convert electricity and CO2 to ethane, because the electricity is not easy to store. But if we convert it into ethane, then that means we are storing electricity in a gas.
Scheller's team is reprogramming these ancient organisms to produce ethane instead of methane, so it can be used in diesel engines.
I mean, the main change is to convert the CC bond. So because the natural methanogens, they convert CO2 to methane. So this is from a 1 carbon to a 1 carbon compound, but it corresponds to an 8 electron reduction. But now if you want to produce ethane, ethane has 2 carbons, so you have to form a CC bond. And the methanogens, they can already produce acetyl-CoA, so this is a C2 unit, but they use that only to grow and not to produce ethane. So what we want to do is to direct the carbon flux fully to this acetyl-CoA, and then actually use this acetyl-CoA to produce ethane from that. And this needs implementation of new enzymes to achieve that, basically. There is different ways to do that. So one way could be enzyme engineering, so to change them from C1 to C2 substrates. But in fact, there are closely related microbes known that can eat ethane and produce CO2. So basically microbes that do the opposite process, they exist. They have been discovered in 2019. And basically we use the same pathway but in the opposite direction. So that means we can actually take the genes from these environmental microbes and implement them in our methanogens to achieve these C2 transformations. So this is actually, I would say, an interesting application of synthetic biology, that you can go to environmental microbes, take their DNA, and then use it to build up a new system, or in our case, the reverse direction of the metabolic pathway. We can have renewable energy converted to electricity, electricity to hydrogen, and then hydrogen and CO2 to C2. But the final goal would be to have C10 or so, like what we use for jet fuels.
But why use such an exotic microbe?
And so instead of just constructing the whole thing always in the same microbe, my approach is to use the microbe that is already very close to the process that we want to make. And then we need not so many steps to do that. Like theoretically, you could try to make the whole ethane project in E. coli, but this will most likely never work. I mean, there's first of all hundreds of genes that you will need to implement, and then you have another redox potential, so the whole cell is not meant for producing methane. But then these archaea, they evolve to produce methane. So for them it should be a smaller step to go from methane to ethane.